Hello students, welcome to the next lecture on bioinformatics. In the previous lecture we learnt about how we are utilizing the latest techniques in biology, latest techniques in other fields like electronics to gather as much data as we can about our environmental microbial communities and other kind of microbes. Now the data that we generate can be up to the order of 60 gigabytes and more, and this is too much data for us to use conventional techniques such as the plain old Excel sheet. So, in order to make sense out of our immense data that we are generating now using next generation sequencing techniques and other techniques, we need to bring in the expertise from different fields, and together we call this synergy between different fields as bioinformatics. So, let us explore bioinformatics. As the name very obviously suggests, bioinformatics consists of two different words, bio and informatics. Here bio hints at the biological origin of our sample, and the informatics here constitutes of information technology and data analytics, which together help us make sense out of our data. Now when we come to the bio part, here we can have genetic sequences, or maybe we can go for metabolomics or metaproteomics, or any of the other advanced techniques that help us gather tremendous amount of data for our biological sample. Now as I mentioned that this amount, when we do these techniques, the kind of information we receive are simple text files usually. Now with this text file we are not really getting information about what is going on in our environmental microbial communities. For example, a long text file that is very hard to open on a simple laptop that has just ATGC repeated all over again in different combinations and permutations does not make sense to an average environmental engineer or scientist. So in order to make sense out of it, in order to be able to answer questions that we have asked in our research, we should be able to use the techniques given by informatics. So this exercise together is called as bioinformatics. Now when we look at bioinformatics, let us look at the definition, the official definition of the bioinformatics. It says here, conceptualizing biology in terms of molecules and then applying informatics techniques from maths, computer science and statistics to understand and organize the information associated with these molecules on a large scale is called bioinformatics. So let us break down this really long definition into small different phrases and try to understand what it is saying. The very first phrase that we ought to notice is conceptualizing biology. So we have biological data that we have generated using chemical reaction, using electronics, or using optical measurements, or just measuring the pH changes as the sequencing proceeds. We have gathered some data. Now we need to understand what phenomena, what environmental phenomena or biological phenomena is being represented by this data. This part is conceptualizing biology. This is going back to our original research question, and we want to understand it in terms of molecules. Now this could be your genetic material, and it could be your metabolites, could be your proteins. So it depends whether you are doing metaproteomics, transcriptomics or genomics, or whether we are going for metabolomics. So once we have this data on our molecules, we can apply informatics techniques from maths, computer science and statistics. So let us say I have a lot of data that I have generated from metabolomics or metagenomics. Then the next step comes is to do quality control on it, QA QC, to make sure that the data that has more error or the data that I cannot be very sure of is removed. Now for this we require certain algorithms that we have from mathematics. For example, we have algorithms that can actually predict what the next nucleotide should be in a sequence, and if it is not that nucleotide which it has predicted, then the chances of it being a chimera, which is basically a false constructed sequence, is higher, and in that case the sequence can be removed. So in this we use the tools from mathematics that they have, the mathematicians who have developed amazing algorithms to predict the genetic sequences. And how do we do that? We do it by using computers, high performance computing more often than not, and here is where we require some programming skills. So typically bioinformaticians are pretty good at basic Bash, they are pretty good at Python, and just general, and it so happens that if you're good at one programming language, others are very easy to catch up. So just general programming. So once you can feed the program in your computer can apply the algorithms, predict which sequences are more likely to be chimera, which are more likely to be a true representative of the sequence that is present in the environment. You can eliminate the chimeras and you can keep the good ones, and you can do other kind of quality control tests too. For example, if there's a repetitiveness, we can remove that also. So now I have cleaned my data. I have even used my programs to align the data with existing database, and I sort of have given it annotation. What is meant by annotation here is that the sequence now has been cleaned and it has been given a name. So I can call it, let's say this is cell 48 sequence. So cell 48 is specific to cellulose degradation. So I can say that this sequence represents the ability to do cellulose degradation, particularly a particular step, rate limiting step of cellulose degradation. So once I have done this data, this analysis, once I have done the annotation, the next step to answer my research question is to do sound statistical analysis for it. Now when we talk of statistical analysis, this is basically testing the hypothesis. We want to find out what is the probability that whatever claim we are making is not a fluke. Whatever observation that we have had is not a fluke, is not by chance, but it is a real, reliable and reproducible phenomena that we can observe over and over again. So this is done by using statistical techniques and tools, and if you are into environmental science, environmental engineering or biology, bioinformatics, I highly recommend you to get in touch with either a good statistician or maybe a good statistics course on NPTEL, and understand at least the basics of statistics, because without this, no matter how good your experimental design is, no matter how expensive techniques you have used to gather good data, and even the good informatics techniques you have used to from computer science you have used to clean your data and make sense out of it, without a backing from statistics, you cannot really have you cannot make any solid claim. So in our environmental science, when we generate data such as genetic sequences, information on metabolites, information on proteins, and we apply techniques from mathematics, from computer science, and even statistics, now together this process is referred to as bioinformatics. And now the word here is large scale. If we already were doing this on a small scale, for example, if I have my wastewater treatment, lab scale wastewater treatment reactors, and I am working on them, so I am doing basic statistics, I am gathering basic data, I am trying to make sense out of it. This is not bioinformatics per se, because it requires that this data generation and collection is done on a large scale. So basically anything that is high throughput and you are analyzing it, you are stepping into bioinformatics. Now another thing I want to mention, that bioinformatics also includes, apart from sequencing and annotating them, bioinformatics also includes modeling. So let us say I know I have identified a protein, its sequence, and now I want to understand what its 3D conformation is. In that case, what I can do is I can use my computational techniques here, and I can make 3D models of the protein, which may be able to predict to some degree of certainty what the function of the protein is, and that I can then test in my lab. All right. So let us again revisit bioinformatics. So what is bioinformatics? What kind of tools do we require? Bioinformatics will give us the tools and algorithms that we need to handle large and complex biological information. This is the definition given by NCBI. NCBI, by the way, is the largest bank of nucleotides. No, not the nucleotides per se, but the largest bank of nucleotide sequences in the entire world, and it is often the gold standard in environmental microbiology and other kinds of microbiology. Bioinformatics is the field of science in which biology, computer science and IT merge into a single discipline, which really brings me up to a very important point that I would like to make here. Often in our colleges, often in our studies, whether we are in undergraduate classes or whether we are doing post-graduation, our expertise is divided into very finely defined narrow disciplines. For example, this is environmental engineering, this is transportation engineering, this is chemical engineering, right? This is physics, this is a chemistry problem, this is a biology problem. Even within biology, we have divided into different groups. This is general biology, this is microbiology, this is molecular biology, and so on and so forth. However, one thing we need to recognize that nature in itself does not follow these narrow definitions of biology, of disciplines. What it does follow is its own nature. So the disciplines are man-made, and now what science is progressing towards is removing the boundaries and going from within discipline research. So this is defined as I, within discipline. So you go in depth within a discipline, to transdiscipline research, where we actually take experts from different disciplines, they come together and they try to solve a problem. This is more like what bioinformatics is. We have computer science, we have mathematics, we have biology, and this is and then we have multidiscipline research, and then we go to interdisciplinary research. So in interdisciplinary research, we are actually not only are people expert in their own disciplines, but their idea of expertise is fluid and it moves through multiple disciplines. So for example, we might have a biologist whose real expertise lies in how to write programs to make sense of biological data. So for example, if you are a computer engineer or computer engineering student and you are part of this course, this is an opportunity for you to use your understanding of environmental microbiology and apply it in your computer engineering to develop new tools and do research there, to make advancements there. And we are also moving towards transdisciplinary research, where we do not define a problem as an environmental microbiology problem or as a computer engineering problem. It's just a problem, and we use whatever tools are available without any constraint of what discipline it is. So to me, bioinformatics is really the cutting edge of moving from within discipline research to transdisciplinary research. And you would not be surprised to know this, I hope, that bioinformatics is where a lot of research is happening across the globe, and there is a lot of demand for it, because most of the bioinformatic processes require a lot of high performance computing, and any kind of algorithm that can reduce the computational cost would be a major demand anywhere. Alrighty, now let's move on to why do we use bioinformatics, or what do we use it for? We use it for storing, retrieving biological information from databases. For example, I have generated a lot of information, and I want to make sure that it is uploaded on a database, it's made public, but manually uploading every sequence would be very painful. So because I have millions of sequences. So by using bioinformatics, I can write commands that will upload it for me, that will give metadata to other users who might be interested in the data. And also, let's say there's a database or there's some information available on MGRaster or another platform, and I want to download it and I want to make sense out of it, then it can also retrieve biological information for me. The other way we use bioinformatics in the same sense of retrieving is, let's say somebody in Austria-Hungary or in Austria sequenced a pure culture and found out the importance of a particular gene, and that sequence is available on a database. They uploaded it on, let's say, NCBI, and they wrote information about it, like what is the speciality of this gene. So I can match the genes that I have sequenced in India with those databases, and then if there is a similarity, I'll get information. Alrighty, this gene that I have sequenced in India matches the gene that was sequenced in Austria, so matches really well. So there's a very high chance that the function of that gene is similar to the function of the gene that I have here. And in this way, I actually retrieve biological information. I can compare my sequence, I can get an idea of how unique or how similar my sequence is, and how unique or similar its function might be. So the other option that we have is that if you're not if you don't want to compare with gene, but you have a protein sequence, what you can do is you can translate a protein sequence, and you will have at least the sequence of the protein, okay, amino acid A follows amino acid B, and so on and so forth. Now in this case, we can do three-dimensional modeling using bioinformatics to find out what would be the secondary structure of your amino acid sequence, and what will be your tertiary structure of your amino acid sequence, what will be its 3D conformation, and then it can match it and predict what kind of properties this protein will have. Alright, next what we can use bioinformatics for is searching for previously known functions of a gene. So for example, I have a gene and I know it is doing some job that it is doing, it serves my purpose, but I also want to know what other people have searched before. So basically this is a very quick literature review for me. I can match this gene with databases, or I can translate this gene into protein in silico, which means I do it on a software, I do it on a computer, I don't have to actually clone it into a bacteria using a vector and then make the bacteria express it. So once I have in silico translated it, I can match it with the existing database and find out what is the experience, what are the observations of other researchers. That's it. I can compare data with other researchers, and then I can compile and distribute data for other researchers. And this is very fantastic because recently with metagenomics we have generated so much data that so let's say, let me start here again. So let's say we have a big part and we have a big world here. Let's say there was some study done in USA and about soil microbiology, and they observed some kind of microbial community, some kind of interaction, and all is well and good. Now let's say some other study, big study that happened in Europe, and they observed same soil microbiology, they found different communities, different characteristics and stuff. Another study in India, another study in Egypt, and then let's say another study in South Africa. So all these studies have collected data from different parts of the world. The one big possibility that bioinformatics gives us is that we can collect data if the data has been uploaded on databases. We can collect this data from different researchers and then do a kind of meta-analysis to understand what are the global patterns. And there have been some really good papers that people have published to see the global patterns of antimicrobial resistance, which is a major environmental challenge. So this is compiling and distributing data for the researchers. Now let's come back to central dogma. So our central dogma, if you remember, is we have DNA which stores data, we have RNA which is messenger basically, and thymine is replaced by uracil, and then RNA is expressed into protein. Now at each of these steps we can do different kinds of analyses. If we are sequencing genome, a DNA, we get genomic sequences, for example metagenomics or just Sanger sequencing. If you are doing for RNA, you can find out the RNA function, you can find out RNA structure. If you are doing at protein level, you can do a lot of work. You can do protein sequence, you can find out what the sequence is, the amino acid, then you can do 3D modeling and try to understand what the structure would be. Again, the 3D modeling of protein sequence to understand its primary structure, secondary structure, tertiary structure, quaternary structure. Now all these and predict its function, all this is part of your bioinformatics, by the way, modeling in bioinformatics. And then you can also do phenotyping from your data. So at all levels of the central dogma you can get data, and we need bioinformatics to do different kinds of analyses. Now again, as a revision of what you have already studied, the basic tools used in bioinformatics most commonly for sequence retrieval to find out the sequence matches are NCBI, GenBank, and other internet resources that I'll be talking about in the next lectures. For sequence comparisons, most commonly used is BLAST, GCG, or MAC vector. For protein, we have some RASMOL, Protein Explorer, and many more. If you log into NCBI page, this is how it will look like to you. And there'll be an icon, welcome to NCBI page, and then now look at the 6 different options that they have on their home. You can deposit data or manuscript into NCBI databases. So let's say I did a study in India and now I have collected some sequences. I mean, I have sequenced some samples and now I have sequences, and I can deposit those sequences. Now there are twofold benefits of depositing sequence to GenBank or to NCBI. To GenBank, because nowadays most journals will ask you, have you deposited your sequence somewhere? And the reason is because we want more easy availability of sequences and we want more transparency in our research. So the sequences, if you deposit in NCBI, you can say, yes, I deposited in NCBI or in GenBank, this is my accession number, this is my annotation, and then you can publish it in your manuscript. And if you have published a manuscript, you can deposit your manuscript. So if anybody matches their sequence to your sequence, in this case my sequence, then they will know, okay, the sequence was found in this kind of environment and this was the study, here's the manuscript of the study that was working on it. So there's an option for submitting, and then you can also download. This is very important. So what NCBI does is that if you send a request to NCBI through internet, it will take your request. Okay, now you are in waiting, and there are many other users across the world that have submitted their request, and one by sequence, NCBI processors, their high performance computing centers will compute the queries, will solve it and give them the data. So there is a waiting time, but if you have a good computer in your house, in your lab, in your room, then what you can do is you can download the NCBI database, and using very simple Linux based bash commands, or whatever platform you are using, Linux, Unix, Windows, very simple commands you can use to actually to align data, to do all kind of analyses that NCBI can help you do. So if you download the NCBI database, you can do a lot of analyses, and this is the beauty of NCBI, that it is open, it's very transparent. Next is, if you have any questions, you want to learn more, and I highly encourage you explore this part if you are at all intrigued by bioinformatics, at all intrigued by the latest advantages and advances in environmental microbiology, is to explore the learn portion. It will help you find documents, attend classes, watch tutorials. So for example, you have a genetic sequence and you want to align it and find out what bacteria it is, and or if you don't know exact bacteria, then what kind of bacteria it might be and where else has it been found. For example, I remember when I was working on oil spill in Gulf of Mexico for my master's thesis, I got some sequences via Sanger sequencing, and not only was I interested in knowing what what microbes are present, but I also wanted to know, were these microbes found in other oil reservoirs or areas where there were oil spills or not, because if they were, then I can say, all right, these microbes might be associated to oil spills, oil reservoirs. So if you want to do this kind of analysis and you're not sure how to go about it, then you can click to the learn option and learn more about it, and we'll be briefly discussing this in our next lectures. And then what you can do is you can actually use in this develop, you can use NCBI API and build your own applications. So let's say you want to do some kind of analysis that is not available here, or you have a cool idea of what kind of analysis would be helpful for the researchers, you can jump into this and code it. And then this is very important, let's say you are not very clear about what kind of analysis to do, you click analyze, it will guide you step by step. And then if you want to collaborate with NCBI, let's say you are a bioinformatics company or you're a bioinformatician, then you can collaborate with NCBI, explore research and collaborative projects here. And this is the web page of NCBI, and the homework for this week actually involves you going to NCBI page, aligning the sequence that I'll give you, annotating it, finding the percent match similarity, then translating the sequence and then running the alignment again. So once you'll be using BLAST N or BLAST X depending on the sequence I give you. In the other case, you'll be using BLAST P for protein. And then you have to compare how the data is different in either case. So please bookmark this slide and make sure that you visit NCBI, familiarize yourself with it, and then thus you can do good in your homework. Alrighty, let's look at similarity searching. So the reason why we use NCBI most often is for similarity searching. We want to find out what is our sequence most similar to, and we can do it for gene sequence, we can do it for protein sequence. And when we align the alignments between the query sequence and any given database sequence, allowing for mismatches and gaps indicate their degree of similarity. So this is the beauty of NCBI, it allows mismatches and gaps. So for example, let's say I have a sequence here, this is your database sequence, okay, database sequence, and if I don't allow for gap, let's say it's starting here and my actual sequence is starting here, if I don't allow for gap, then I might have lot of mismatches, does not match, these are regions that don't match. But if I allow for gap and I shift it, okay, if I shift the same thing here, then there is a very solid match. So this kind of gaps and skips and misses are permitted in NCBI to account for the possibility that your clone or where you fragmented your DNA to is different from where the other people who have submitted to database fragmented their DNA from. Already, the structure, function and evolution of a gene may be determined by such comparisons. So if you can read the manuscripts of people who have submitted their genes, if you can read about it and know about it, you can get an idea of function, you can get an idea of structure, you can also get an idea of evolution, how is the gene changing, because remember, to understand evolution, we need to know how the genetic sequence is changing. So if you have genetic sequences, you can align them on conserved region. This we talked about in last class. So let's say these are your genetic sequences from different times or different regions, and there is a conserved domain. So you align it on the conserved domain, and then you see how are changes happening here. And then this way you will know what is the rate of evolution of this particular gene under given circumstances. Alrighty, algorithms. So computer comparisons using mathematical probability. So you have some input of data, and then we give you some output, and the way the mathematical algorithms work is that they find the best matching pair when comparing the sequences. So basically there are two sequences that they're comparing. They'll compare it by allowing all kind of mismatches, all kind of gaps. So maybe this is how it would look like. Maybe it will look like this, or maybe it will look like this. So we are giving a gap here, we're giving a gap here, we're giving gap in between. So it tries for all permutation combination and then tells you what is the best match scenario. Alrighty, so let's look at these two cases. So here we have two sequences and we want to find out the match. So if you look at here, we notice that there is a 70% match. We are allowing for these gaps. Now if you can translate, now this is genes. So I hope you can recognize that C stands for cytosine, A stands for adenine, T for thymine, and so on, same thing. And T matches with T, A, T, G, A, T. So 7 out of 10 are matching, so you have 70% match. Now if you translate this into protein, so you read it like this and then you translate it into protein, then this is what you have. So here you are doing blast N or blast X, and here you are doing blast B. So you are comparing the proteins, and then you notice that the match percentage is much higher. Why would this be so? Because if you remember from your amino acids, different codons can code for same amino acid. So to account for this degeneracy, we prefer that you translate it into protein and then match. You get a better similarity and also a better representation of what is perhaps happening, because the degeneracy in the genetic sequence will not impact the functioning of cell. It will not impact the functioning of protein, because the protein is right, protein is the same. So blast P is very useful. And now let's look at sequence analysis. First you determine the correct sequence of DNA, then you compare the sequence with known sequences, you translate DNA into protein sequence, and then you understand protein function in context of all the data you have gathered here. Alrighty, the current tools used in bioinformatics have certain strength. They are very accessible. So most of the tools are freely available online. In fact, if it is proprietary, it tends to not have a big market. So people are making their tools, their databases available freely to everybody. They are growing very rapidly. They are very user friendly, and I must add that they have very good forums where you can ask for help, you can post questions, and then they have very good community support here. Most of them have very good community support. What are the weaknesses of current tools? Well, sometimes they're not up to date because some of them can be really obsolete. It has limited possibilities, and one of the limitation we talked about last time was there's not a single tool that can do all kind of comparisons that someone might be interested. And then we have limited comparisons and information, and many of them are not accurate. They have statistical errors, we have issues there. So this is, I like to think of this as room for improvement. Bioinformatics as it is right now is not set in stone. It is growing very rapidly, as it says here, and thus we are evolving very fast when it comes to bioinformatics. So this is where the room for improvement is, and this is where people are working to improve our tools. So need for improved tools for Human Genome Project, gene array technology, comparative genomics, and functional genomics. For this we need specialized sophisticated tools. For proteomics we need to understand global view of protein function. For example, there might be protein A, and in one particular cell it does a particular function and the cell can survive, but you put it in another cell and then it is not acting in isolation. It is acting in presence of many other inhibitors and many other molecules, many other proteins, many other RNAs. So its behavior in different cells will be different. So it's very important for us to understand the global view of protein function and not just say, well, it matches to this protein found in this bacteria, so it must be doing the exactly same thing. And similarly for protein motifs and for structural databases. So basically bioinformatics allows us to do data mining. It allows us to handle enormous amount of data. It makes it approachable and acceptable. It helps us understand what is important, what is not, because now here's another thing. In metagenomics, there's a rough estimate that 80% of your data, so 80% of your base pairs would be housekeeping genes. So 80% of genes that you will detect from metagenomics will be housekeeping genes which are found in every bacteria. So you won't really see much difference. You won't really see the trend that you're looking for. So only the meaningful data for you would be 20%. So bioinformatics will help you find out what your 20% is, where it is, and then you can do all kind of statistical tools. You can use them, you can do all kind of analyses to answer your questions, to test your hypotheses. It also allows us to manipulate and analyze data to find patterns and variations that correlate with biological function. There's another added part of this bioinformatics that I would like to mention briefly here is data visualization. Most of the tools, bioinformatics tools nowadays, they come with very good inbuilt data visualization techniques, and if we do not have data visualization techniques in the tool, then they will recommend you, why don't you use package A, software B for visualizing your data. And this is very important because we have so much data but we cannot really make sense of it unless we can see it, as humans are very visual creatures. So besides all this, we also have the portion where we are using bioinformatics to visualize the data. And in the next one of the next lectures, I'll be talking about I'll be comparing two software platforms. One is Mother and the other is Chime. So it is written as QIME, pronounced as Chime. And I'll be discussing how each of their advantages and what their disadvantages are. And one of the advantage for one of them is that they have very good data visualization. So dear friends, this is all for today. And in the next lectures, we will be actually looking into these tools, and I'll be briefly telling you, briefly leading you on how to use them. Thank you.